Thank you so much, everyone. It fills my heart with joy unspeakable to talk to you all on promising molecular targets and the role of next-gen sequencing and early phase drug development in colorectal cancer. Again, I'm Vivek Subbiah. I'm available on Twitter and Blue Sky. These are my links. So we are in an era of precision oncology, right? We moved away from diagnosis from light microscope, and we currently live in an era of molecular microscope. What do I mean by molecular microscope? The next-gen sequencing. 20 years ago, they said we cannot sequence, the human genome. Then it took $3 billion to sequence the human genome. And right now, we can get it done, in commercially available agent for a few hundred to few $1,000s, depending upon where you are. So we currently live in an era of molecular microscope. So what are we looking forward in the future? We are looking into an era, the future era of multimodal fusion, moving on to an AI era microscope, where AI can help us with amalgamating and assimilating all the data that we have, not just medical records, genomics, proteomics, spatial transcriptomics, and everything to help us move forward. We are evolving to an era of tissue-agnostic drug development and tissue-agnostic therapies in precision oncology. We are moving away from one-size-fits-all chemotherapy to histology-dependent targeted therapy to tissue-agnostic therapy. So there was this commentary in Nature that said, forget lung, breast, prostate cancer, or colorectal cancer. Why tumor naming needs to change? The conventional way of classifying metastatic cancers according to the origin is denying and delaying access, people access to drugs that could help them. So we are in an era of tissue agnostic therapies where we are thinking differently about cancers. We are thinking about the targets, not the tumor. And colorectal cancer has also benefited from this evolution of tissue agnostic therapies, where we are moving away from the organ to the histopathology, which are histology agnostic or histology dependent. So what is a tumor agnostic therapy? Tumor agnostic therapy are used to treat any kind of cancer. So we are used to be thinking like breast cancer, kidney cancer, prostate cancer, or colorectal cancer. But tumor agnostic therapy treats the cancer, as long as the cancer has the underlying molecular alteration and or biomarker. So what about the current landscape of tissue-agnostic precision therapies? Again, today, we have around 10 different drugs approved in a tissue-agnostic manner. Again, for pembrolizumab, starting from pembrolizumab for dMMR positive cancers, 3 drugs for NTRK fusion positive cancers, larotrectinib, entrectinib, and repotrectinib. Again, dabrafenib and trametinib for BRAF mutant positive cancers, selpercatinib for RET fusion positive cancers, and Enhertu or trastuzumab deruxtecan for IHC 3+ positive cancers. So this is tissue agnostic therapies across all diseases. What about colorectal cancer? So colorectal cancer has been molecularly profiled deeply and has been divided into different molecular subtypes, like the conventional pathway, the serrated pathway, the alternative pathway, and the other pathways, based on the molecular alterations like non-MSI high, MSI high, or SIMP low negative, BRAF wild type, KRAS mutated, and other markers. But what influences the treatment choices in metastatic CRC today in December 2024? The tailored patient choices are mainly based on patient characteristics, tumor characteristics. Slowly, molecular characteristics are being incorporated, like the RAS, BRAF, MSI high, and HER2. Of course, we do patient-centric therapies and patient-centric drug development. The patient preference is also important that influences the treatment choices in CRC. So currently, what is the metastatic colorectal cancer treatment flow? Again, after first-line, second-line therapies, you can see a smattering of molecular alterations like HER2 amplification, MSI high, dMMR, BRAF V600E. Let's talk about them. Let's also finish this presentation by talking about emerging therapies in colorectal cancer. Again, what are the clinical studies that we have for molecularly targeted areas in colorectal cancer? Let's talk about the Beacon trial with encorafenib, binimetinib, cetuximab for BRAF V600E cancer. Then talk about the Breakwater, which is a combination of chemotherapy plus BRAF plus EGFR therapy, and talk about the MMR therapy, MMRD, MSI high and dMMR therapy, and the Mountaineer and the Destiny study for HER2 positive colorectal cancer. Again, what is a beacon study? The beacon study is encorafenib, which is a BRAF V600 inhibitor, in combination with cetuximab, which is EGFR monoclonal antibody, plus or minus binimetinib, which is a MEK inhibitor in BRAF V600 mutant CRC. As you all know, BRAF V600 alterations occurs in 10 to 15% of patients with colorectal cancer and confers a poor prognosis. In the Beacon study, the CRC safety lead-in consisted of the triplet regimen and had manageable safety profile. Here shows the primary overall survival and objective response rate of encorafenib plus cetuximab plus or minus binimetinib in BRAF V600E mutant colorectal cancer. This shows the updated overall analysis and overall survival, and the triplet and doublet demonstrated improved overall survival and objective response rates in patients with BRAF V600 mutant metastatic colorectal cancer when compared to current standard of care. So currently, we have encorafenib plus cetuximab, which is FDA approved for BRAF V600E positive CRC, and it is NCCN guidelines. It's second line plus with cetuximab or panitumumab. Breakwater. So what is a breakwater? This is BRAF plus EGFR targeted therapy in combination with chemotherapy, taking it to the next level for first or second line BRAF V600 mutant CRC. As you can clearly see with these waterfall plots, clearly the combination is active. This shows the progression-free survival by blinded independent central review. Moving on to the MSI high and DMMR colorectal cancer. Again, this has been moved to frontline. This shows the EP-Nivo versus the standard of care chemotherapy. This shows the progression-free survival of EP-Nivo. So most recently, hot off the press, as of last week, nivolumab plus ipilimumab in colorectal cancer was published in the New England Journal of Medicine. Clearly, you can see the progression-free survival outcomes was significantly better with nivolumab plus ipilimumab than with chemotherapy. Again, clear benefit with hazard ratio. Again, even the grade 3, grade 4 adverse events occur less often with the immunotherapy than with the standard of care chemotherapy. So checkpoint inhibitor therapy is now indicated in first-line treatment of DMMR and microsite satellite unstable high metastatic colorectal cancer. In addition, pembrolizumab is also FDA approved for first-line treatment. What about HER2 targeted therapies? Again, this is a Mountaineer study. So what is a Mountaineer study? Tucatinib plus trastuzumab versus tucatinib in HER2 mutant, HER2 positive CRC. Again, what is tucatinib? It's a tyrosine kinase selective inhibitor for HER2. And the dual inhibition with a tyrosine kinase and a monoclonal antibody with other agents can have a deep inhibition of the HER2 pathway. This shows the waterfall plot and change in tumor site. The objective response rate was 38.1%, and the duration of response was 12.4 months. So what does it show? Right? Tucatinib in combination with trastuzumab has a potential to become new standard of care for patients with HER2 positive metastatic CRC. An ongoing phase 3 study is comparing tucatinib plus trastuzumab plus chemotherapy with modified 446 with standard of care. HER2 targeted therapies, we have a host of HER2 targeted therapies that include HER2 monoclonal antibodies, HER2 antibody drug conjugates like Enhertu, trastuzumab emtansine, trastuzumab deruxtecan, and also we have emerging HER2 bispecific antibodies like zanidatamab, which was recently approved in the US in an accelerated approval manner for HER2 3+ biliary tract cancers, and several other bispecifics in clinical trial development. As you can see, the HER2 targeted therapies, we have a broad array of HER2 targeted therapies in early phase and phase 2 studies. And this area is one of the hottest areas in oncology drug development. And we have the advent of HER2 CAR T-cell therapies, HER2 NK cell therapies. So I think, we have a host of targeted therapies for HER2 pathway directed therapy. So what about Destiny? Destiny is the study with trastuzumab deruxtecan, which is Enhertu. Again, this compared two dose levels, 5.4 milligrams Q3 weeks and 6.4 milligrams. And interestingly, as you can see here, the objective response rates was 45% in the 5.4 milligrams and 27.5 in the 6.4 milligrams. Again, this shows the median overall survival with the 5.4 milligrams and 6.4. Again, clearly promising activity is observed with Enhertu in HER2 positive metastatic CRC at both the dose levels. Numerically higher objective response rate was seen at the lower dose level, which is a 5.4 mg per kg, than with 6.4 mg per kg. And the greater anti-tumor activity was seen with HER2 IHC3+, which the response rate was 46.9%, than with IHC2+ at the 5.4 milligram dose. So again, it's hard to compare two trials, apples with apples, but comparing both the Mountaineer study combines a TKI plus monoclonal antibody, and this is an antibody drug conjugate, and interestingly similar objective response rates. And we really look forward for better and better targeted therapies, and we have great options for patients with HER2 positive cancer. What about tissue agnostic therapies in this space, right? So we have NTRK fusions. NTRK fusions are seen in colorectal cancer as well. And in that data set, the primary data set, there were 4 patients with colorectal cancer and 2 achieved PR. So the objective response rate in colorectal cancer harboring NTRK fusions with larotrectinib was 50%. And with entrectinib, the objective response rate was 20%. There were 10 patients with colorectal cancer being treated in that cohort. And interestingly, with RET fusions, the objective response rate was 20%. So as colorectal cancers also harbor RET fusion, and the colorectal cancer objective response rate was 20%. So what about the emerging targeted therapies in CRC? We are really looking forward for antibody drug conjugates. As you all know, we live in an era of antibody drug conjugates, and we have interesting antibody drug conjugates that are active in metastatic colorectal cancer. Again, this is AbbVie 400, which is a c-Met antibody drug conjugate with a TOP1 payload. Again, clearly, as you can see here, there is activity in colorectal cancer. Activity is observed across all doses in third line plus metastatic colorectal cancer. Again, there are several registration studies going on in colorectal cancer. With this agent, we look forward for more data with this drug in the future. Another antibody drug conjugate is anti-CEACAM5 ADC with the TOP1 payload. And interestingly, CRC also has CEACAM5 expression. And this was presented earlier in ASCO this year, showing early signals of activity in metastatic CRC. And hopefully, we'll see some more data in the next phases of trials with this drug. What about KRAS? Again, KRAS G12C in colorectal cancer, we saw some data earlier this year with adagrasib plus cetuximab. Again, clearly, showing signals of activity. The confirmed objective response rate was 34%, and disease control was observed in 85.1%. Again, this has the potential to become a new standard of care in KRAS G12C positive colorectal cancer. Again, clearly showing progression-free survival of 6.9 months and median overall survival of 15.9 months. What about KRAS? Again, beyond the KRAS G12C, I think we, in 2024 and moving on to 2025 and 26, we are going to be seeing a host of agents going after all the KRAS alterations beyond KRAS G12C. KRAS G12D, which is seen in colorectal cancer, KRAS G12V, KRAS G12R, among others. We are seeing a lot of agents, as you can see here. And we are like ready, set, go, setting off on the mission to target KRAS in colorectal cancer. It's one of the exciting times to do KRAS drug development in colorectal cancer and other cancers. And we have different agents with different mechanisms of actions that include pan-KRAS, pan-RAS, and KRAS G12D specific drugs, and mutant selective KRAS specific drugs, and PROTACs that can degrade the RAS. And it is very exciting to be in KRAS drug development. So we mentioned mainly about genomics, and this is a story of 6 blind scientists and cancer research in colorectal cancer. Again, we spoke about immunogenomics with MSI-High, genomics with BRAF V600E and KRAS. But beyond all genomics and immunogenomics, we need to think about metabolomics, transcriptomics, microbiomics, and proteomics, and have a comprehensive molecular portrait of every patient with metastatic colorectal cancer walking in the door. So in summary, All patients with colorectal cancer need molecular testing. Universal genomic testing that includes germline testing is needed to win the battle against metastatic colorectal cancer. The available currently molecular targeted therapies are for MSI high, BRAF, HER2, and of course KRAS G12C, emerging therapies for KRAS G12D and pan-KRAS. What is exciting in colorectal cancer, we are seeing antibody drug conjugates targeting c-Met and c-Cam are promising. Hopefully, the ongoing studies may bring options to patients to earlier lines of therapy.
Don't forget to like, comment, share, and subscribe to OncoDaily on YouTube. Hit the bell icon to stay updated.